Hi, so this chapter is about osmoregulation, which has to do with water balance, electrolyte balance, and also getting rid of nitrogenous wastes, which is what excretion is. And in vertebrates, this has to do a lot with the kidney, and there are some other structures in other animals that do the same thing. So osmoregulation, this is controlling water and the concentrations of different osmotically active molecules. So osmotically active molecules or solutes, these would be all of the salts, also other hydrophilic molecules, including things like glucose and amino acids, would also be osmotically active. So excretion is getting rid of nitrogenous waste and metabolites, and these are a result of protein metabolism. So when we break down protein to use for energy or break down protein to make new proteins, that nitrogen component of the protein, which initially is in the form of ammonia, NH3, cannot be reused by animals. It has to be gotten rid of in some way. And that's really what excretion is about. For aquatic animals, this is quite easy. They have all the water available to them that they need to flush away waste products that they don't want. It's a completely different matter for land vertebrates specifically, and we'll talk about those situations. So osmoregulation is about taking in water to the tissues, releasing water from the tissues, and also taking up and releasing solutes across the membranes. So remember that to get across a membrane, the membrane has a hydrophobic interior layer. Let me get my pointer here. Membranes have a hydrophobic interior layer, and the only way that anything hydrophilic is going to cross that membrane is by being transported across that membrane. Or it has to have some kind of a channel protein. So water gets across membranes using a channel protein, because even water is hydrophilic. Water cannot cross membranes without having a channel protein. Pretty much every cell has a channel protein, though, with one exception in the kidney that we'll talk about. So usually what we're talking about is the movement of solutes across membranes, and then that is driving water movement across membranes, because water will flow by osmosis if you move the solutes from one side of the membrane to the other. So osmolarity is related to tonicity, that concept that we learned last semester. So if the concentration of all the osmotically active particles in a solution, that's the osmolarity of the solution. And this is kind of a specialty word for biology, because in chemistry there's possibly other solvents and other kinds of solutions, but in biology we're talking about things dissolved in water, and that's pretty much it. And often the only thing that's going to cross the membrane is going to be water. Ions and other osmotically active molecules are either being transported across the membrane or sometimes being moved across the membrane, but water can usually always move across the membrane. So and water is always going to go from a high concentration of water to a low concentration of water. And so that's going to be from an area where there's low solute concentration to an area where there's high solute concentration. So the particles that are dissolved in the water, those are the solutes. Water is the solvent, and the two of them together are the solution. So if they are isoosmotic, that means that there's the same number of particles dissolved on both sides of the membrane in the water. So water's going to move back and forth, but it's already in equilibrium if they're isoosmotic. Hypoosmotic would be less concentrated in terms of the number of dissolved particles. Hyperosmotic means a high number of dissolved particles. So in a hypoosmotic solution, that means the water concentration is high because there's fewer dissolved particles. For hyperosmotic, that means we have lots of dissolved particles, so the water concentration is actually going to be low in that solution. And water is going to go from a high water concentration to a low water concentration, and that's why water moves from a hypoosmotic environment to a hyperosmotic environment, because in a hyperosmotic environment, that means there's less water, more dissolved particles. So if we have two solutions here on either side of a selectively permeable membrane, on this side here, this is the hypoosmotic side, but that means that there's a higher concentration of water on that side of the membrane. If water's the only thing that can cross the membrane, water is going to flow from a high concentration to a low concentration, so that means water is going to flow this way. So osmoregulation presents a problem for animals, and it's a different problem depending on if they're a marine animal that's living in salt water, if they are a freshwater animal that's living in very dilute, mostly water, something that's very hypoosmotic, or if they're in brackish water, or if they move back and forth between different concentrations of water, or if they're on the land. Because all the cells in an animal's body are bathed in a watery solution. And that concentration, for animals especially, the concentration of dissolved particles in that solution that's bathing all the cells of the body can't vary that much, or it's going to damage the cells and interfere with processes in the cells. So regulating the osmotic, or the osmolarity of the interstitial fluid, that's really what we're talking about. Keeping that at a steady concentration. And animals that regulate that, which would pretty much include all the land animals and all the freshwater animals and even some marine animals, these are osmoregulators. So osmoregulators have to expend energy to maintain homeostasis for water and solute balance in their bodies. And sometimes these could be organisms that live in a hyperosmotic environment, like bony fish, or in a hypoosmotic environment, like freshwater animals are living in a hypoosmotic environment. Osmoconformers, these are the kind of animals that you would expect to be osmoconformers. They're invertebrates. These are things like the jellyfish that just don't have any way at all to maintain a difference in solute concentration between the fluid between their cells and their outside environment. They don't try to regulate their osmotic balance at all. Most animals are what we call stenohaline, which means that they're adapted to, and this is stenohaline, we're talking about things that are aquatic. They can't tolerate substantial changes. If you take a freshwater fish and put it in a marine environment, or you take a marine fish and you put it in a freshwater environment, they're going to die. They can't tolerate a big change in their external osmolarity. They're adapted for a particular environment. There are some organisms, however, that are urohaline, and these can survive. Doesn't matter what the external osmotic environment is, and the example here would be salmon, and also eels would fall into this category. Organisms that live part of their life in a marine environment and part of their life in a freshwater environment. They can tolerate huge fluctuations in external osmolarity. That's more in the unusual situation. So most marine animals, the simple ones, all the invertebrates pretty much, are osmoconformers, meaning that they expend no energy to try to maintain a difference in osmolarity between their interstitial fluid and the surrounding seawater. They are whatever whatever osmolarity the seawater is, that's what their interstitial fluid is as well. They're not trying to maintain a difference between that. Vertebrates, however, including fish, are going to be osmoregulators, though, and they are actually hypoosmotic to the seawater, meaning that seawater has more dissolved particles in it than the interstitial fluid of bony fishes. And they have to balance that. They're drinking large amounts of seawater, and that's going to give them excess salt. So bony fish that are marine animals eliminate the extra salt through their gills and their kidneys. And they do have a primitive kind of a kidney that doesn't, it's not, fish have a kidney that doesn't really try to preserve water in the body. Because, you know, they live in water. Water is not they don't have a shortage of water in their environment. Freshwater animals, however, have a problem because even single-celled protists have a problem that live in freshwater, and that problem is water is constantly going to be coming in by osmosis because they live in a very hypoosmotic environment, even to their single cell if it's a protist. So they're going to lose salts as well, and drinking all of that fresh water is not going to replenish all of the salts that they need. So instead, they're going to have to maintain their salts and not lose salts through their gills or through their kidneys the way that marine fish do. So here's a marine fish, and we can see things that are coming and going from the body here. Water is the light blue, salt is the dark blue. So marine fish, remember marine means seawater, so it's quite salty. Their body is hypoosmotic to the seawater, which means they need to take in more water than salt. They're going to have excess salt to deal with. So they're drinking water and salt all the time as they eat. They're taking in water and salt. The way that they oxygenate their blood is by taking in water through their mouth into their pharynx and then flushing it out through their gills. So they're constantly taking in a lot of salt and water into their bodies. So they actually can excrete salt from their gills because they're getting rid of excess salt. They will also lose excess water that way, and they can excrete sodium chloride and other minerals from their kidneys, and they actually don't produce very much urine because they need to keep that water internally. A freshwater fish has a different problem because the water that they take in with food and through just passing water over their gills, water is constantly going to be entering their body, and not a lot of salts are going to be entering their body. They're going to get some of these electrolyte ions through their food, but not as much as they need. So they can do the opposite with their gills. They can actually take up the small amount of salt that's found in the water that they live in and take it up and actively transport it across the cell membranes into their bloodstream. They're also going to excrete huge amounts of urine to get rid of all this excess water that they're constantly taking in, and that's how they can maintain their balance. There's also another weird adaptation because, you know, in biology there's always something weird. And these are animals that can allow themselves to be completely dehydrated. And specifically, we're talking about tardigrades. Remember, they're their own phylum. They're very weird. There are marine tardigrades, but most of them live on land. They live in very wet environments normally, like in mosses. One of their nicknames is moss bears. And they can allow themselves to be completely dehydrated into an inactive state, and this is called anhydrobiosis, because they're still alive. Anhydro means without water, so they're still alive, but completely dehydrated, and then when they become rehydrated, they wake back up again. And this is actually a really weird adaptation. There aren't very many animals at all that can go through a long-term dormant state like tardigrades can. Land animals generally, as we talked about before, have a problem with water loss, just like plants did when plants first adapted to the land. Animals, when animals first adapted to the land, one of the main problems is reducing water loss from the surface of the body. And we talked about arthropods having this hard outer coating in their marine environment. That gave them a little bit of an advantage in trying to adapt to the land because they already had this hard outer coating that would have them have less water loss. Well, having a way to maintain water balance while getting rid of nitrogenous waste is also a problem. And many land animals, including insects, have evolved ways to conserve water by not having to lose a lot of water in urine. So what kind of nitrogenous waste they get rid of, and remember nitrogenous waste is mostly from protein metabolism. There's also some from breaking down DNA and RNA. But most of it's from breaking down amino acids, using excess amino acids for energy. And the simplest method for getting rid of nitrogenous waste is not to change it at all. When its proteins are broken down, you get ammonia as that amino group gets broken off of the amino acids. Ammonia has no stored energy. It has no potential energy. It's just a waste product that needs to be gotten rid of. It's pretty toxic and has to be diluted in a lot of water. So this is the molecule that animals are getting rid of that live in marine and freshwater environments that are osmoconformers. So there are two other forms, though, that are ways of making that nitrogenous waste more concentrated and conserving water. These both require energy, though. Ammonia is the product as it's initially made. Animals that get rid of their nitrogenous waste in the form of urea or uric acid, however, that takes energy to convert them into those forms. However, urea and uric acid are less toxic, and so you don't need as much water. And so that's why we see these adaptations in land animals. So ammonia is the initial waste product that's made. Even in our bodies, when we're breaking down amino acids and using them as energy, ammonia is the initial product that's made. Very much of it cannot build up, however, or it becomes toxic to cells. So animals that live in an aquatic environment, though, they don't have to worry about that, especially something like a nematode here, which is not having any concerns about toxins building up in its body. It can just excrete them. And so it wastes no energy at all changing ammonia into anything else. It just excretes it by diffusion across the whole surface of its body into the surroundings. And that's what most invertebrates do. Urea is less toxic, and so that means it can be concentrated. If you have your nitrogenous waste in a molecule that's less toxic, that means that you can have urine that's more concentrated, you can conserve more water, and you don't have this toxic compound in the body. In vertebrates, urea is usually produced in the liver, carried by the circulatory system to the kidneys, and then it is excreted. And we'll talk about that process that occurs. So making ammonia into urea takes energy, but we can have it as a much more concentrated product and thus save water. Uric acid is even more along those lines. So uric acid really can be excreted as a paste. You can have it even more concentrated than urea. It's even less toxic than urea, and so this allows for the best conservation of water. So organisms that excrete uric acid as their nitrogenous waste includes insects, land snails, reptiles, birds, because because they only have a single opening, their cloaca, the uric acid can just mix with the feces on the way out, and that's the white stuff that's in bird poop. That's the uric acid. So they poop it out. The uric acid along with their feces come out right together, and very low water loss. So here's our three groups here. So we have breaking down of proteins into amino acids. We lose an amino group, also a smaller percentage of it from nucleic acids and like RNA and DNA. Breaking those down, you also end up with excess amino groups. This is the structure of urea. That's what we excrete. Mammals excrete urea. And uric acid, a little bit more energetic to produce, but allows much greater conservation of water. Birds, reptiles, insects, they barely excrete any water at all. It's a really great conservation method. We can excrete urea in fairly concentrated form, but we still need some water to get rid of it. Ammonia requires lots of water to get rid of it. So aquatic animals excrete ammonia. So the excretory systems are about tubules, and filtration from blood and interstitial fluid into and from the coelomic cavity in some cases for the simpler animals, and then filtering that and getting rid of it. That's really what the excretory system is about. So here are the four processes that we're going to talk about in more detail, especially as regards to the mammal kidney. And the four steps are filtration, and this is the fluids leaving the bloodstream and going into the tubule. Reabsorption, reabsorbing the valuable nutrients like glucose and amino acids that filter out. Secretion, actively transporting across the membrane things that are waste products. And then excretion, processing the filtrate that now contains nitrogenous waste and getting rid of it out of the body. So these are the four processes. So as we're talking about the different parts of the nephron, which is the tubule in the kidney, think about which of these four processes is occurring in each bit, because it's often more than one is occurring in each bit. Some of them there's only one, but for most of the tubule, there's at least two of these processes that occur in that particular section of the nephron. So this is what we're going to be talking about. This is a very simplified tubule. So here we have filtration from the blood. That's step one. So we have a fluid here. There's cells don't cross. Large molecules like proteins in the blood don't cross and aren't filtered out, but small molecules in water all filter out. So glucose, amino acids, salts, water that's in the blood, all of that filters out and ends up in the filtrate. Reabsorption occurs because we want to preserve all the amino acids and the glucose. We don't want to be excreting those in our urine, so those are reabsorbed. A lot of the salts are also reabsorbed. Secretion is also an active transport. So reabsorption, there's a lot of active transport going on here. The same with secretion. active transport across the membrane of things that we don't want. And then excretion is this substance leaving the body with the nitrogenous waste plus some other things. Excess salts, excess carbonate ions, excess water. Okay, so that's what we'll talk about in the next video, is all of the different types of kidneys.